So the first talk is to be given by Dr. Tahmeed Ahmed, who's a very prolific nutrition pediatrics related scientist from the ICDDRB. And he's got a whole lot of accolades to his name. So he is, from the point of view of this symposium, he is known very much for his microbiota-directed complementary food as an intervention for childhood malnutrition. Dr. Ahmed, please.
Good afternoon, sir. Distinguished chairs. First of all, I would like to thank Professor Ganguly and Dr. Hajela for the very kind invitation. I had wanted to be in person to attend this very important meeting and to learn myself, but unfortunately I had to be on duty travel, and I am now in Nevada, and it's 2:35 a.m. in the morning. So, distinguished chair, please allow me to share my slides. I hope you can see my slides and you can hear me. Can you hear me?
Yes, sir. Yes, sir.
Okay.
Please
Okay.
continue.
Thank you. Thank you. So again, thank you for giving me the opportunity. And I would like to talk about microbiota-directed food in the treatment of childhood acute malnutrition. Over the next 30 minutes or so, I would like to discuss about the role of gut microbiota in a very formidable problem, which is malnutrition. And then I would like to discuss about how we have developed and put into use the microbiota-directed complementary food in improving wasting or acute malnutrition among children. So first of all, acute malnutrition. As I talk to you, you might wonder what is the number one and the most common ailment or disease that afflicts children all over the world. This is malnutrition. And today we have close to 150 million children under the age of 5 years suffering from stunting, which is a chronic form of malnutrition. And we have 45 million children who are suffering from acute malnutrition, which is also known as wasting. And this is such a great irony because the world produces so much food, and yet we have to see this huge burden of this malnutrition. This is a photograph that I had to take in one of the rural centers in Bangladesh. And this is a very typical photograph of any of low and middle income countries, including India. Some of these mothers will have children who are suffering from acute wasting. And as I said that we have more than 45 million children, if you look at the demographic divide, only about 25% happen to be in Africa. And look at the burden in Asia. 70% of the total burden of acute malnutrition comes from countries located in Asia, particularly South Asia. And these are the global nutrition targets about childhood stunting, childhood overweight, anemia, breastfeeding, low birth weight, and then at the right lower corner, we have childhood wasting. The goal is to reduce and maintain childhood wasting to less than 5%. Unfortunately, in many of the countries that we are talking about, including mine, rates of wasting are pretty high. The latest survey that we have done in Bangladesh gives a prevalence estimate of 11%, and that translates into about 1.7 million children suffering from acute malnutrition. What is happening to the rest of the world? We can see that there are only 29% of the countries that are on track to achieve the target by 2025. Rest of the countries, more than half of them, they are off course. And that's why it's very important to find out new treatments for the management of childhood acute malnutrition. Back in 2015, 2016, I, along with my dear collaborator, Dr. Jeffrey Gordon of Washington University, we proposed this hypothesis. If you have a mother who follows all the norms during pregnancy and then gives birth to a child by normal vaginal delivery, the child is breastfed, the mother doesn't take any excess courses of antibiotics, the child gets a normal dose of the gut microbiota from the mother. There is no perturbation, and the child has an endowment of age-appropriate gut microbiome, which is mature and has normal physiological, metabolic, immune, and neurological development. On the other hand, if the mother has a C-section, which is very common in Bangladesh, if the mother has to be treated with courses of antibiotics, if the child is not breastfed, then you can see that the gut microbiota becomes disturbed. And this disturbance leads to impaired microbiota maturation, which has huge consequences. Back in the early 2000s, we tried to first look at what happens in children with acute malnutrition. We are doing a trial of a treatment in children with severe acute malnutrition, and we thought that first we should try to see what happens to the gut microbiota in healthy children who are living in slums of Dhaka city. We then compared the gut microbiota of healthy children living in slums with children suffering from severe acute malnutrition, either treated with a ready-to-use therapeutic food or a local diet composed of khichdi and halwa. The very colorful columns that you see on the left, that denotes the gut microbiota of the healthy children living in the slums of Dhaka city. On the other hand, the two very dark columns denote the sparsity of the gut microbiota, and these are from the children who are being treated for severe acute malnutrition. We could also demonstrate back in 2014 that there is profound gut microbiota immaturity of children suffering from acute malnutrition. This was done using 16S ribosomal RNA sequencing and published in Nature in 2014. And we thought that there were several options. One could be FMT, which we have heard about this morning, this afternoon. And then we also thought that a more feasible option would be a food-based intervention that could control this gut dysbiosis. We then thought that first we should know whether this is a cause or an effect of malnutrition. So we took the gut microbiota from children in Dhaka, children who are malnourished in the hospital, and children who are healthy living in slums of Dhaka city. We gave this gut microbiota endowment to mice, and these are the notobiotic mice, mice that are born and brought up in a totally germ-free environment in the laboratory of Dr. Jeff Gordon in St. Louis, United States. The mice also receive the food that is consumed by healthy children living in slums of Dhaka city. And then we looked at the weight of these mice. In this slide, you can see the red graph, and this red graph indicates mice that receive the gut microbiota from healthy children living in Dhaka city. And you can see a static representation of the weight of the mice. On the other hand, mice that received gut microbiota from children suffering from acute malnutrition whom we were treating in Dhaka, they had a dramatic fall in their body weights. And this suggested that this reduction in body weight, in other words, malnutrition, is a cause of the faulty gut microbiota that they received from children endowment. So we then postulated whether gut microbiota immaturity is an effect or a cause of undernutrition. We did translational studies, and we found that immature gut microbiota transmit features associated with childhood malnutrition to recipient mice. We also found from these mice experiments that impaired growth, metabolism, immune function result, and we tried to identify growth-promoting bacterial strains that are underperforming in the immature microbiota. And then with this knowledge, we thought that we should develop microbiota-directed therapeutic candidates using these animal models. So modulating the gut microbiota to improve growth in children, a problem that is so profound, affecting millions and millions of children all over the world. So we thought that how could we increase the numbers of good growth-promoting bacteria in this intestine? One correct way would be to promote their numbers by giving them an appropriate diet, because like us, the gut bacteria, they also need food for their sustenance. So this is then what we did. We did some nutritional anthropology in the slums of Dhaka city, and we identified the different food ingredients that are used by moms to prepare complementary foods. And you can see on this slide, green bananas, chickpeas, eggs, milk powder, rice, spinach, sweet pumpkin, tilapia fish, whatnot. Based on this commonly used food ingredients, we developed several microbiota-directed complementary foods, 1 to 19. For example, if you look at microbiota-directed complementary food number 4, that would contain a preponderance of green banana. It would also contain chickpeas, egg, and then at the bottom, it would contain spinach. So these were the MDCFs that we developed, and then we wanted to test them in animals as well as in children suffering from acute malnutrition. In animals, in notobiotic mice, we tried to give these foods to mice, and we tried to look at what happens to the gut microbiota. So this is a correlation matrix, and you can see if you focus on the red squares, and these red squares indicate the strong positive correlation. A green banana, chickpeas, tilapia, peanuts, these are all very positively correlated with some of the very important growth-promoting microbiota members in the intestine. For example, Faecalibacterium rossniesii, Ruminococcus storks, and then Doria longicatena. So we identified the food ingredients that were able to replenish the good members of the gut microbiota. We also identified which are the good members of the gut microbiota that can promote growth in children. So with this, we went on to identify the 3 most prominent microbiota-directed complementary foods. And first of all, we tried to look at a pre-proof of concept study. This was a small study, a little more than 74 children, again in the slums of Dhaka city, and they were suffering from acute malnutrition. A control was also given, a ready-to-use supplementary food, which is made of rice and lentils. This is one of the MDCFs. We call it MDCF2 because in this pre-proof of concept study, MDCF2 actually stood out as the first candidate. And this is prepared from chickpea flour, soybean flour, peanut paste, green banana powder, soybean oil, sugar, and then the micronutrient mix. At the bottom, you can see a mom. She is feeding her child in one of the nutrition centers established in this slum. At the top of the slide, you can see the prepared MDCF very much looking like halwa, which is very common in our countries. And this is the composition of the study diets. On the left, you can see ready-to-use supplementary food, where you have protein, fat, carbohydrate, fibers. What is important to note here is at the bottom, you can see that MDCF2 has about 20% less calories compared to ready-to-use supplementary food. From this proof of concept study, 1 month study, we found that yes, it does repair the microbiota immaturity. It promotes some weight gain. It increases plasma biomarkers. It leads to an improvement in bone formation, development of the CNS, as well as immune function. But this was a pilot study, not powered to look at the clinical efficacy. So we then went on to look at the efficacy of this microbiota-directed complementary food. This was again a trial in the community. The main objectives included promotion of repair of microbiota immaturity, promoting the proliferation of beneficial bacteria in the gut, improving both ponderal as well as linear growth in children, and finally, improving the metabolomic profile of children suffering from moderate acute malnutrition. Study outcomes included, among others, weight for length Z-score, which is a marker of wasting, weight for age Z-score, mid-upper arm circumference. We also wanted to look at what happens to linear growth, which is measured by length for age Z-score. We did this somologic scan by which more than 5,000 plasma proteins could be assessed and profiled. And finally, the gut microbiota configuration as a result of the 16S ribosomal sequencing. And we tried to look at amplicon sequence variants. Here is the scheme of the trial that we did. First week, pre-intervention, then the 3 months treatment phase, followed by 1 month of post-intervention. There are 2 arms, one arm MDCF2 and the other arm rice lentil-based ready-to-use supplementary food. At different points of the trial, the children, we took the samples, blood and stool samples from the children. This is the environment where we did the trial in a slum of Dhaka city. In this efficacy trial, there were 124 children, about 62 in each arm. They were 12 to 18 months old and suffering from moderate acute malnutrition. For the purpose of the trial, we prepared the ready-to-use supplementary food as well as the MDCF right in a kitchen that was established in the middle of this slum. So just to give an idea about the baseline characteristics of the children, there was no difference. Exclusive breastfeeding was observed in only about 25% of the children. Let me share with you the results. The results are important. At the top, you can see the anthropometric features at baseline. In the middle, this is the mean rate of growth per week during the 3 months of intervention. And you can see the highlighted areas. This is the difference between RUSF and MDCF2. And this is positively different between the 2 arms. At the end of the 1 month follow-up period, which is 4 months, in addition to seeing a significant difference in weight for length Z-score as well as weight for age Z-score, we also found a significant difference in mid-upper arm circumference. Another way of looking at the improvement in weight for length Z-score is this. You look at the graph and you can see the blue line represents MDCF2. This is the weekly increase in weight of the children, while the red line indicates, represents RUSF. The difference in change is 0.011. What does this mean in clinical medicine, pediatrics? With this difference, over a period of 1 year, you would expect a child to have a growth to the extent of 0.57 compared to RUSF in favor of MDCF2. We also tried to look at what happens to the plasma proteins. And this was done, as I said, by somologic scan. And then we used a special technique called gene set enrichment analysis. Through this technique, we had an idea about the proteins that could be ordered according to how they promoted the expression of a particular phenotype. And you can see on the left-hand side of this slide that there was a significant change in weight for length Z-score and the protein levels that we observed. On the right-hand side of this slide, you can see positive association of the plasma proteins with weight for length Z-score. And these proteins were associated with ossification of the long bones, development of the brain, development of cartilage, endochondral bone morphogenesis, while on the other hand, you can see the blue bars that represent the plasma proteins that had a negative influence. For example, these were associated with viral transcription. These were also associated with, interferon signaling pathway. What we found was that MDCF2 was associated with more proteins that were positively associated with weight for length as well as all the positive phenotypes, for example, ossification. On the other hand, RUSF was associated with more of the blue indicated proteins that had a negative influence on weight for length as well as being associated with all the wrong phenotypes. Another way of expressing this, at the top you can see the proteins that were associated more with development of the CNS, more with MDCF2 than with RUSF. At the bottom, the same that denotes with the development of CNS development, and the top one is for ossification of bones. What happens to the gut microbiota? We found that weight for length Z-score was positively associated with a proportion of growth-promoting members of the gut microbiota. In total, we found 209 specific taxa or bacteria, and out of this 209, 23 had significant association with weight for length Z-score. Of these 23, 21 denoted here by the red bars, these were positively associated. And these indicated the members of the gut microbiota, which we now know as being very important for growth of children. For example, Prevotella, and then we have Faecalibacterium prausnitzii. But at the bottom, you can see two members of the gut microbiota which were negatively associated. These happen to be Bifidobacterium species as well as our friend Escherichia coli. So this was the finding, summary of the findings. We have developed this MDCF2, which was able to repair immature microbiota, promote weight gain, and increase plasma biomarkers related to bone formation, neurodevelopment, and immune function. We also found that MDCF2 was linked with 70 plasma proteins and 21 bacterial taxa that were positively correlated with weight for length Z-score. The rate of change in weight for length Z-score as well as weight for age were more observed in children who received MDCF2 compared to the control RUSF. This is an article that was published several months in The Economist about the microbiota-directed complementary food. So what is the way forward? Right now, there is a large trial. This is actually a multi-country trial taking place under the auspices of the World Health Organization in Bangladesh, India, Pakistan, and 2 other countries in Tanzania as well as Mali. On the other hand, we have also improved this MDCF2 to being a product that conforms with the UNICEF and WHO specifications, the Codex compliance. And this would be a product that can be used in different programs, taking care of both children with both severe acute as well as moderate acute malnutrition. And this is the team at ICDDRB, and I would like to thank you again for giving me this wonderful opportunity to be present at this meeting. Thank you very much.
Thank you, Professor Ahmed, for a wonderful talk. So I think we can have the discussion right now itself because it is 2:45 a.m. There, maybe 3:30 now. So we should not push him to wait for the end of the session. So please.
Dr. Tahmeed, thank you very much for coming at this early hour, and you made the day here. This was a nutrition-based meeting, and thank you for giving this wonderful talk. I just have a small comment that two things which bothered us in a low weight with babies and those who didn't have linear growth, how to save their brain and how to make their bones strong, because ultimately the metabolism of these children were totally skewed, and these two were the major victims. Your food supplements actually are giving proteomic signatures which ossify the bones, make them stronger, and also build up the brain. So neuromimetic peptides. So this is wonderful. And the other thing which comment which I want to make, because this we are now very sure, and you have taken all the three parameters of the growth of the children. But one of the things which I want to ask you a question, that bifidobacterium, why it did have a negative thing? Because bifidobacterium brevi is very important for children. I couldn't understand. But one of the things which you have found, I have also found in one of my studies, Doria. Doria is a Parsonizi, and Doria are the two very important gut microbiome, which changes metabolism favorably. But my question is to you that why the bifidobacterium had a negative correlation.
Thank you very much, sir. When Professor Ganguly asked me to attend this meeting, I could not say no, even at this very early hours of the day. And thank you very much, sir, for leading ICDDRB when you were member of the Board of Trustee of ICDDRB. So let me try to answer your first observation about low birth weight babies. Indeed, we should also have a food like this for low birth weight babies, but unfortunately, this is a food that is based on, macronutrients, carbohydrates, complex carbohydrates that we can't give to these babies. These babies would benefit best from mother's breast milk. But we now believe that, this endowment from the mother is very important. And we have already observed the presence of enteropathy in women of childbearing age in Bangladesh. And we believe, and we have proved that the cause of this enteropathy is also gut dysbiosis. Sir, we are now working on experiments in Dhaka by which we are going to probably see this gut dysbiosis, and we are also going to identify members of the gut microbiota that are at fault. Now, this could be corrected in mothers so that when the child is born, the child is going to have a healthy endowment of the mother's gut microbiota. I think this would be very important for the very young and tender child for suffering from low birth weight. And that is why, as I speak to you, I must also say that side by side, taking care of childhood malnutrition, it's extremely important that we also pay a lot of attention to maternal undernutrition, which is also so profoundly common in the countries where we live. Bifidobacterium, this is a very interesting character. If you look at the gut microbiota of healthy children, you will find, particularly babies who are breastfed, you will see that there is preponderance of Bifidobacterium infantis. These are the members of the gut microbiota that thrive on human milk oligosaccharides coming from mother's breast milk. As the child grows old, the mother's breast milk reduces, and so does the preponderance of Bifidobacterium infantis. I think that is one of the reasons why we saw a negative correlation of Bifidobacterium, because these were children 12 to 18 months old.
Yeah. Can I? Yeah, Dr. Ahmed, this is Dr. Jagmeet Madan. I'm a professor from SVT College of Home Science, SNDT Women's University, Mumbai. And I'm also the National President of Indian Diabetic Association. I think it was a privilege to hear you, sir, and the elegant work which you presented. It excites me as a nutritionist, and I know that how challenging it is to do community-based studies. So from your experience, sir, we would like to understand what have been your learnings and challenges, in doing this kind or this scale of community-based studies, and especially when we know that in developing countries, it's not only feeding the children, there are, comorbidities which come with lack of sanitation, hygiene, all these confounding factors are also there. So what are your learnings, if you could share with us? And also, I would be very happy if you could share with us what is the work which we are doing now in these different countries together in this direction. Thank you, sir.
Thank you very much. Several questions. First of all, how do we do this type of work in the community? I'm sure that you have the same experience that we have. It's sometimes becomes quite a lot of challenge, but the most important thing is to establish a very good rapport with the community. And the slums where we work, they know us and they think that we are friends, not foes. And before each study, I must repeat, madam, before each study we do in the community, we have a meeting with the community elites. And these community elites include the school teachers, the primary school teacher, the fathers of children living there, the shopkeepers, the religious leaders. And like I have presented to you with a PowerPoint slides, we also have these PowerPoint presentations for them. And we give them a cup of tea and they ask questions. These are all very searching questions which we have to answer and satisfy them. So this is very, very important. Another challenge for doing this type of studies is sample collection. These are the things that we use for DNA sequencing. Now we have progressed from DNA 16S to much more sophisticated types of DNA sequencing. These all require collection of biological material as soon as it is passed. So we have our health workers who literally go and collect these stool samples within 20 minutes of passage and place it in liquid nitrogen. So this is again, this is a very good experience. And with this experience, you can do it better day by day. You mentioned about water sanitation hygiene. Again, so important in our geographies. And again, we have shown that the duodenum of children suffering from environmental enteric dysfunction, this is supposed to be sterile, but we have found that as many as 14 different members of the gut microbiota, they inhabit the duodenum of these children. And this is all a result of the poor sanitation and hygiene. Maybe open defecation is not a huge problem, but still mothers, they don't practice hand hygiene. And this is so important for preparing this complementary food. You asked about what we are doing. I mentioned about these 5 countries. This is called the NutriMam trial. where we are giving this microbiota-directed complementary food to the mothers. They prepare it at home and they're giving it to the children. And the control is the locally available food. In different countries, we have different food, and this is a trial in progress. Hopefully, in the next 18 months, we'll have the results from this trial. There are also several other studies in progress, and some are in plan in Bangladesh.
Thank you.
So Dr. Lahiri from India, a pediatrician, absolutely wonderful. So I hope to meet you actually this time. But I think
you.
I have a question. I mean, it's actually, I wanted to ask, have your advice, is that we have the horizontal and the vertical gene transfers, which happens from the mother to the child. We're already starting with a vulnerable population of malnourished mothers. And the diversity and the quality of the microbiota in the mother is already jeopardized. Now, would decades of malnutrition be covered up with this kind of a therapy? And would probiotics really help? Because we are talking of giving probiotics to the mother in the second and third trimester and to the baby. But would it really make a big difference? Just your comments on this. Thank you so much.
Thank you. There is a slight difference between probiotics and this type of intervention. Probiotics are usually aimed at improving general health. On the other hand, what we are doing is, biologically active substances. And in this case, we are trying to modulate the gut microbiota. And there are specific mechanism of action. For example, why does MDCF2 work? What is there? And we have found these are things that were published last month. Because of time, I didn't want to include that. We have found that it is not the protein, rather it is the carbohydrate in the MDCF that contains specific glycans. And these glycans include mannan, arabinose, and several other substances. These are taken up by these particular growth-promoting bacteria. These are the substances that help this bacteria to proliferate, and in return, they do all the good things in the body of the child.
Thank you, Professor Tahmeed Ahmed. So we will end your session now. So we wish you a happy good night.
Thank you so much.
So,
thank you.
Thank you for the wonderful presentation.